Hello, in this video we will discuss about the insulin, a way of production as well as the synthesis of the insulin. We will discuss in this video. So here is the person which that is contain a digestive system. During the salivary glands produce the saliva during the eating time. So when we will eat the carbohydrate, starch, sugar and protein, so the starch will be semi-digest into the oral cavity through a salivary glands. So these salivary glands which that is convert into the amylopectin into the amylose. So this is due to the amylase enzyme. So for example, here is the after the digestion complete in the duodenum, that is the simple sugar will be formed as well as the simple amino acid. So this glucose will be enter into the bloodstream directly, but the fat is also present in the form of globule and in the form of droplets. So these droplets will be enter first into the lymphatic system and then after go into the bloodstream. But here we will just understand and concentrate on the amino acid as well as the glucose for giving the response through a pancreatic insulin. So the beta cell of pancreas will be producing insulin. So here is the pancreas. During the increasing of the glucose level after the absorption in the bloodstream, so will stimulate the pancreas to produce the insulin. This insulin is basically here is the production of insulin, so that will be received by the tissue, and it means the GLUT4 channel will allow to move inside. For example, here is a muscle is also look like that. So the active transport is also, so ion exchanges due to the sodium will be move inside with a one glucose molecule move inside to form a glycolysis to produce pyruvate and ATP, while on the other hand the glycogenesis. So the amino acid is also metabolic pathway which that is allowed to move inside through a insulin basically. So we will not understand about that. So here is the bloodstream when the insulin bind with the GLUT4 channel of the liver. After the binding of the GLUT4 channel with the liver, basically is enter the sugar. So this sugar molecule will be converted in the form of glycogen because the anabolic hormone is the insulin. So acetyl-CoA is also produced, and this acetyl-CoA go into the Krebs cycle basically and to produce also the triacylglycerol, and the triacylglycerol which that is basically packaging as well as the apoprotein through a amino acid basically is to produce a packaging to form a very low density lipoprotein. This lipoprotein contain also cholesterol. So cholesterol is also synthesis through a acetyl-CoA. We will not understand about that here, so I will make a separate video about that. So here is the bloodstream which that is basically very low density lipoprotein will be enter into the bloodstream. So it will be move and provide the free triacylglycerol into the adipose tissue. Here is the adipocytes, basically is the very low density lipoprotein receptor are present which that is bind and to give the triacylglycerol to the adipose tissue. So the adipogenesis basically the lipogenesis will be occur. So this is convert into the low density lipoprotein to high density lipoprotein. It means the protein content will be increased after the donation to the adipocyte or adipose tissue. So that is the basically metabolic pathway which that is overview of the pathway. So here let's begin to understand the mechanism, and we will also understand the synthesis of the insulin also. So here is the pancreas which that is contain a islet of Langerhans. This islet of Langerhans basically contain a alpha cell, beta cell and delta cell. Delta cell which that is regulate the beta cell and alpha cell produce somatostatin, while the beta cell produce the insulin and alpha cell produce the glucagon. So let's begin to understand and concentrate on the alpha cell. So here is the beta cell. So here is the beta cell which that is I am magnifying this beta cell to understand as well. to understand their mechanism. So here is the when blood glucose level will be high, it means the blood vessel contain a high number of glucose as compared to the normal, so that will be trigger the insulin production. So that triggering of the insulin production mechanism we will discuss first here. So let's begin to understand. Suppose the glucose level is high in the bloodstream. So the glucose will be high, it means the GLUT2 channel will allow to move inside of the pancreatic cell without using insulin. So this is the way to more better chance for the production of insulin. So here is the GLUT2 channel. When enter, the glucose will convert into the pyruvate through glycolysis, and after the production of the pyruvate, 2-pyruvate will be produced, and it means it will be go into the mitochondria. After the going into the mitochondria will produce the ATP through a electron transport chain through a Krebs cycle. So that ATP is increased. For example, here is the ATP gated potassium channel. This channel will create the charge inside as compared to the outside difference. So this charge is due to the potassium. So ATP increasing to inhibit the potassium efflux. After the efflux inhibiting, the inside charge will be negative. After the negative charge inside as compared to the outside, it stimulates the calcium channel, L-type channel, which that is create calcium move inside. So the influx. So the calcium move inside, it will play a different function, and it stimulate the insulin insulin granules, which that is stimulate to due to the positive charge are present. Due to the charge increase, basically is the increase of the charge negative, sorry, so the negative charge. So the exocytosis will begin after the stimulation by the calcium ion. So these exocytosis through the calcium channel, and this insulin will be increased in the blood. After the increasing of the insulin, which that is play a different function, basically the binding with the muscle as the liver, and it will bind with the also adipose tissue to move the glucose inside of the adipocyte. So here is it is also stimulate the DNA to increase the gene cluster containing insulin producing gene cluster. So here is a chromosome, and it is produce messenger RNA, and messenger RNA go into the cytoplasm, and the transfer RNA will be bind, and polysome will formation will be occur, and these produce the protein polypeptide. This polypeptide, after the termination through a transfer RNA, that is the codon, which that is for termination is the UGA and UAA, which that is terminal. So here is the messenger RNA. A lot of messenger RNA will be produced again and again to produce a more and more insulin for the exocytosis. Why? Because the blood glucose level is high, so we need the insulin. So we need the lower the blood glucose for moving into the muscle. So that means endoplasmic reticulum will mature this protein to convert this type. So the N terminus and carboxy terminus is present. This pre-pro insulin will convert into the pro-insulin, and this pro-insulin polypeptide will convert into the insulin mature after the going into the Golgi bodies from the cis to trans side. So it means insulin structure is look like this. This is the sulfide disulfide bond bridge making formation to create a two strand of the polypeptide. After the formation is the insulin vesicle will be formed. This vesicle is look like a structure. So here is I am drawing the muscle which contain a tissue. This tissue is the myocyte, and basically is the muscle cell. This muscle cell is, for example, we will discuss how to perceive the insulin. So the tyrosine kinase receptor are present in the form of pair, and this tyrosine receptor, when bind with the insulin, two molecule of the insulin to come together, and autophosphorylation will be occur. After the autophosphorylation, this insulin after the binding will autophosphorylation to phosphorylase the amino acid containing tyrosine. This tyrosine, that is why its name. So here is the extracellular domain and here is the cytoplasmic domain of the tyrosine receptor. Tyrosine kinase receptor, sorry. So kinase means it will be phosphorylase to the tyrosine containing residue. So basically the target protein is here. This target protein is contain a tyrosine amino acid, where the tyrosine are present in the protein will bind with the phosphate group. After the binding through a tyrosine kinase receptor, after the binding basically is stimulate the IRS1. This IRS1 is play important function. For example, growth, gene expression, fat synthesis, protein synthesis, and glycogenesis through a channel creation. So this channel in the form of here, which that is produced by the creation of the channel through a movement of cytoplasm to the plasma membrane. So amino acid will be move inside, fatty acid move inside, and glucose also will move inside. After the movement of the glucose and fatty acid as well as another, so let's begin to understand. This is, suppose this is the liver cell and muscle cell, collectively we can say that. And if glucose will be move inside, it means the glycogenesis will be formed in the liver and the muscle. While on the other hand, if the fat is entered into the liver cell, for example, so it means produce the lipogenesis to produce very low density lipoprotein, as well as the adipose tissue if that adipose tissue are present here. So on the other hand, it is anabolic hormone. It means it produce the anabolism, not catabolism. On the other hand, the growth and gene expression, fat synthesis, protein synthesis, glycogenesis, that is many anabolic pathway which that is lead to insulin receptor tyrosine kinase through produce IRS1. So here the insulin will be increased, so the blood glucose level will be decreased, and glycogen stimulation and glucose to convert glucose 6-phosphate and hexokinase through a hexokinase and glucose transporter channel in liver and muscle will be produced, and inhibition of proteolysis and glucose converting to the acetyl CoA will be also inhibit, and triacylglycerol will be produced, as well as the muscle growth and tissue repair. So I hope you make sense. Insulin is the anabolic hormone which that is produce the anabolism.